When you're talking about antibiotics, resistance is inevitable, right? It's not a question of if, it's a question of when. What we're really focused on at Genentech is treating life-threatening infections that are associated with hospitalized patients.
From the gram-positive bacterial perspective, one of those that's the most familiar to everybody is Staph aureus, or MRSA. It's really one of the poster childs for what happens when bacteria become resistant to antibiotics. It still is an unmet medical need, but gram-negative bacteria are much more difficult to treat. And the reason being is they actually have two-cell membranes.
These are bacteria that are resistant to all of the antibiotics that the medical community currently has at its disposal. So when we started this project, our goal was to introduce an entirely new molecule that the bacteria had never seen before that would be able to treat these highly resistant bacterial infections.
The second membrane is basically the equivalent of a force field. It protects the cell from antibiotics penetrating into its cell interior.
So one of the key challenges for developing any new gram-negative antibiotic is finding ways to get molecules through that force field. Many antibiotic targets actually reside inside the cytosol, in the center of the bacterial cell. So there are a number of reasons that we are attracted to cyclopeptidase as a target. It's an essential protein. The bacteria can't grow without it. It's also relatively accessible. Cyclopeptidase is located on the outer surface of the inner membrane, meaning that molecules only need to cross that outer membrane to reach the target. And finally, we knew that cyclopeptidase is druggable. We knew this because of natural product inhibitors that have been described in the literature previously, such as the aromycins.
And so our efforts were largely focused on essentially driving down and improving the affinity of the molecule. And that's what we did over the past 5 years, is through just a dedicated chemistry effort. We deconstructed the whole natural product.
The program to date has made at least 1,700 analogs. So it's a real testament to the medicinal chemists on the team, to their hard work and their creativity figuring out ways to make these sorts of complex molecules.
And inevitably, we came up with some of our most potent molecules, which are orders of magnitude, probably 10,000, 100,000 more potent than what the natural product aryllomycin is, natively speaking. And throughout our course of modifying the aryllomycin scaffold, we were able to find molecules that indeed penetrate the outer membrane to access the cyclopeptidase.
That was an aha moment for us. We can kill these bugs. But of course, that's not the goal ultimately, right? So with our potent molecules in hand, we reached out to a resource provided by the Center for Disease Control. Now, they've collated a collection of highly multi-drug resistant isolates.
These are some of the most severe pathogens that are out there that are absolutely killing patients.
And it was incredibly gratifying to see that not only do our molecules kill these laboratory strains of E. coli, they also kill highly multi-drug resistant clinical isolates. And that was really the vindication moment for this program. Seeing our molecule's ability to kill a strain of bacteria that is literally resistant to every other approved antibiotic on the market today, that made it all worthwhile. Because it's been almost 50 years since a novel molecule, a novel class of molecules, has been approved to treat gram-negative infections. What we hope is that new therapies, a new class of antibiotics, will ideally buy you a window of time. And so we can never stop developing novel antibiotics because they won't stop evolving resistance.